Hello, I'm Jim Cornell, and this is Base to Base, the weekly biotech podcast from Air Coastal Media. Yes, welcome again to the Base to Base Biotech podcast, and this slightly longer episode is going out on May the 16th, just in time for the Eurovision Song Contest in Switzerland this Saturday, something that I managed to avoid for many years living in North America. I guess I could just not watch, but something makes it compelling, and it's definitely not the music. Maybe there should be a Eurovision biotech contest, where company CEOs from each country do presentations about their latest Phase 3 results, dressed in strange costumes with a pounding beat in the background. If you have suggestions for who should be on the podcast, or have much better ideas than the song contest, which wouldn't be hard, please do get in touch at jim@deeptechdigest.com. One event that did take place recently, completely unrelated to music, is the American Association for Cancer Research, or AACR, annual meeting, which was held in Chicago. And this week's podcast is dedicated to some of the companies that were there. So, this week we have 3 conversations. They are with Matt Robinson, SVP of Research at Microbiotica, Mohan Ottawa, CEO and co-founder of OneCell Diagnostics, and André Blaukat, CEO of Tessellate Bio. The only question, I guess, is in which order. Well, it doesn't matter too much as they're all very interesting conversations. So let's kick things off with Tessellate Bio. The company, which was at AACR, has partnered with Boehringer Ingelheim to develop first-in-class precision treatments for people with hard-to-treat cancers. To tell us about the company and the partnership, and much more, is its CEO, André Blaukat. Maybe to get things started then, I wonder if you could tell me a bit about the origins and the journey so far for Tessellate Bio.
So Tessellate Bio was created at the beginning of the pandemic, of the COVID pandemic in 2020, based on the research of Klaus Assalin, based in Portugal, in Lisbon, and Hilda Pickett, based in Sydney. They have discovered independently a target that is synthetically lethal with cancers that use a specific telomere maintenance pathway called alternative lengthening of telomeres, or ALT or ALT. So this was the starting point. They published this in Nature Communications back-to-back in 2019, and one of our investors from BGB, Daniela Couto, she has seen these publications and said, wow, this is exciting science, and it's published back-to-back in different places of the world, so this must be real. This was the starting point of the company. Yeah, and then a few years later, in end of 2022, I joined the company as a CEO. And yeah, we have been working on this concept of synthetic lethality and expanding the concept of synthetic lethality to new patient populations. The concept of synthetic lethality means that under certain circumstances, cancers or tumor cells that acquire constantly mutations, in some cases create, by acquiring a mutation, a dependency on a pathway or on another gene that is redundant or non-essential in normal cells. But due to their specific mutations, they are dependent on this gene or pathway. And this offers opportunities for pharmacological intervention and treatment. And I think most famous example that has validated this concept are PARP inhibitors in breast cancer, ovarian cancer, pancreatic cancer, and prostate cancer patients that have mutations in a gene called BRCA. And BRCA is a gene that regulates DNA repair pathway called homologous recombination repair. So that's how the company started. And then, we have evolved over time, initiating new projects based on synthetic lethality. And what is unique about our company is that we want to extend this concept beyond the traditional patient population that are targeted by synthetic lethality. And these are mainly patient populations that have a defect in homologous recombination repair, also abbreviated HRD. So most of the PARP inhibitors and most of the concepts that have been initiated in biotech and pharma are targeting this patient population. And that's about 15% of all cancer patients, high unmet medical need, but it's getting more and more crowded in this space. And we believe there's a potential in the 85% of cancer patients that do not have a defect in homologous recombination repair. It's a bit more challenging to identify synthetic lethalities, and the targets are often a bit more challenging, difficult druggability scores and so on. And that's exactly our sweet spot. So exploring new biology and having a slightly different risk appetite with respect to the targets we are attacking.
So how did you approach that when you decided to not go the same way as everybody else? How did you develop what you're doing?
First of all, important for us is a very strong biological validation of the target. And as I mentioned, the fact that two top labs in this telomere biology space have validated the target back to back gave us confidence. And then we confirmed the data internally that by knocking out the target, we see lethality in cancer cells that use the alternative lengthening of telomere pathway, while others that use alternative telomere maintenance pathways and normal cells were not affected at all. And then we quickly did a feasibility screen. So that's something that we are typically doing when you're dealing with challenging targets. Instead of, discussing the challenges and the technical feasibility forth and back, we often do a quick feasibility screen. And if this is positive and we identify early hit matter, we do the full screen and then continue optimization of hits towards leads with in vivo activity.
What kind of cancers are you, or is your approach applicable to?
So the approach is a precision medicine approach that is independent of a specific indication or cancer type. It's more dependent whether patients have either this genetic aberration or the corresponding phenotype. In case of our ALT programs, that they use alternative lengthening of telomeres. And these are 10 to 15% of all cancers. There are mesenchymal cancers like sarcomas, where more than 50% of all cancers use this pathway. Brain cancers as well, but then there are also cancer types with a higher prevalence, like colorectal cancer, small cell lung cancer, where between 1 and 10% of cancer patients this pathway is used. And that's also important. In order to treat these patients, you need to have the right diagnostic to select patients. If a certain biomarker, certain phenotype is only present in 1 to 10%, you need to pre-select patients because your drug won't work in the entire patient population. And this is also something that distinguishes us from many companies. We take this really serious, the development of diagnostics, in this case called companion diagnostic, because they enable an appropriate use of the drug. So we have, whenever we start the drug discovery program, we start in parallel diagnostic development.
It makes sense to work on the two in parallel. Do a lot of companies not do that?
Well, more and more companies are doing this, but I know from my own experience in big pharma that the diagnostic development is often delayed, or investments. And in some cases, it's too late. So then you have a drug, you want to start clinical trials, but your diagnostic is not either biomarker is not appropriately validated or the diagnostic is not ready to go. And then you do an all-comer phase 1 trial, unselected patient populations, and you miss corresponding signals. And in many cases, do not, leverage the full value of the drug for patients. We take this very serious and make sure we have the right diagnostic in place before we start clinical trials.
What's your ALT program and how does that work?
As I mentioned, ALT stands for alternative lengthening of telomeres. Telomeres are the ends of chromosomes. And during every cell division, they are shortened. And this is actually an intrinsic clock of cells that limits the replicative lifespan. So it limits the number of cell divisions a cell can undergo, because if they reach a critical length, the cells don't divide anymore. They either die or they become senescent, meaning, they stay there and do their job. Cancer cells have to overcome this, because one of the hallmarks of cancer is that they have an unlimited replicative potential. So they can divide in an unlimited fashion. So cancer cells must overcome this with no exception. And there are only two pathways how they can do this. So nature only offers two pathways. One is reactivation of an enzyme that is called telomerase. So it can elongate telomeres. Has been a popular target in the pharma and biotech industry. 30 years of research, a single drug approved in a very small indication. And we believe, and many others believe, that the reason for this is simply the lag phase between the pharmacological inhibition and the desired biological outcome, so killing of cancer cells that use this mechanism. We are targeting the pathway which is called alternative lengthening of telomeres, which is used by 10 to 15% of all cancers. And this is a different mechanism. So this pathway relies on homologous recombination, and cancer cells that use this pathway, they have to pay a price, because excessive use of homologous recombination leads to replication stress. And this replication stress leads to can lead to DNA damage and a strong DNA damage response signal. And cancer cells that use this mechanism have to balance this. And our scientific founders have identified the target or the enzyme complex that is balancing this replication stress. And they have shown, we have replicated this, when you delete this target or pharmacologically inhibit the target, you selectively kill cancers that use alternative lengthening of telomeres.
And does that affect normal cells or just the cancer cells?
It selectively affects cancer cells that depend on alternative lengthening of telomere. No effect on normal cells and no effect on cells that use telomerase to maintain their telomeres.
And you also have another program, the LOF program?
LOF stands for loss of function. And here we are targeting loss of function of a tumor suppressor called RB1. One of our scientific collaboration partners has identified synthetic lethality in cancers that have a loss of RB1. And the target here is SCIP2. It's part of an E3 ubiquitin ligase complex. So one of the functions of RB1 is to keep SCIP2 in check. SCIP2 is an E3 ubiquitin ligase, meaning that it ubiquitinates proteins that are then degraded. And one of the targets of SCIP2 is an endogenous cell cycle inhibitor called P27. And in tumors that lack RB1, P27 is excessively degraded. And this allows these cells to quickly proliferate. So our goal is to block the interaction between SCIP2 and P27 and thereby prevent degradation, ubiquitination and degradation, an accumulation of P27, and allow this endogenous cell cycle inhibitor to do its job. And this is stopping cell proliferation.
Where are you at on the clinical trial path?
Yeah, we are still a bit away from clinical trials. So we have identified promising hit matter that we are now expanding and preparing for in vivo experiments this year. I should say that the target is well known. I mean, the biology is super well validated, but the target has been considered as undruggable because the interaction between P27 and SCIP2, it's a very flat surface, no really druggable pockets, at least if you look into the crystal structure. What we have done is we have applied state-of-the-art molecular dynamics. So trying to model the dynamic of the protein and not only looking at the static crystal structure. And in this dynamic studies, we have seen evidence for potential cryptic pockets that open up during a certain time or during certain dynamics of the protein. And this was a starting point for our screen. And as I mentioned earlier, we didn't discuss the feasibility for a year, but we said, okay, let's do a feasibility screen. Let's invest a bit of money. Let's do a screen. Let's see if we can find binders to these cryptic pockets. And, I wouldn't talk about this if we weren't successful with this. And meanwhile, we have crystal structures of our compounds with SCIP2 really proving that this cryptic hypothesis is true. And the compounds do what we expect them to do, preventing binding of P27 and increasing P27 levels, stopping proliferation of corresponding cancer cells and driving them into cell death.
When it comes down to the patient at the end that has cancer or any disease that this is applicable to, are you aiming to extend life, or are you intending to make it something that's manageable in the same way that HIV is today, or are you looking to stop it completely?
Yeah, our vision is to turn cancer patients into cancer survivors. In an ideal case, of course, we want to cure cancer, and this is a very ambitious goal, but I think it would be already a significant step forward to manage cancer, a bit like you described for HIV patients, to convert it into a chronic disease that can be very well managed.
You're not there yet, but how would you be administering this drug, or is it too early to say?
So we are developing small molecules that are orally available. So we will provide or administer a pill. We do expect in selected patient populations monotherapy activity, so that it works as a single agent. But we also know that in oncology cancer treatment, it's usually combinations that are most efficacious and give the best benefit for patients. So therefore, we are also early on exploring rational combinations for our drugs.
Are you able to tell me anything about the partnership with Boehringer Ingelheim?
So this is an exclusive license for our ALT program. We received an upfront payment that I cannot reveal in detail here, R&D funding, milestones, royalties, so a typical licensing deal. It's a collaboration, with a stepwise transition of activities to Boehringer Ingelheim. So at the beginning, we work jointly on this, or we will do the majority of the work, and then stepwise transition it to Boehringer Ingelheim, who will then, at the end, conduct clinical trials.
Very good. What else do you have in your pipeline? As if that wasn't already enough what you're doing now, but
yeah, the key priority for us is, of course, to make this collaboration with Boehringer Ingelheim successful and then bring our fully owned RB1 loss of function program to the clinic and to a significant value inflection point and a point where it can really make a difference for cancer patients that suffer from cancers with RB1 loss of function. We also have a couple of early ideas. We have established a bioinformatics platform called Tessellate Analysis Engine that is integrating external data, public data, but also internal data. And our bioinformaticians have developed algorithms to identify new synthetic letalities, new targets that we are validating in the lab, but we haven't started another full drug discovery program yet, but have plenty of ideas should we be able to raise enough money to implement these ideas.
Those are the only questions that I had. Is there anything that you wanted to talk about that we hadn't covered?
What I didn't mention is, I mentioned initially that the company was created during the pandemic, with a footprint all over Europe, so people from different places in Europe. But since 2 years, we have own labs in Stevenage at the SBC. So it's halfway between Cambridge and London. Very well-equipped biotech incubator, closely associated to one of the biggest R&D sites of GSK. So a great place to do research.
Well, if tumours can be stopped or killed, that is great news. And it's wonderful that so many companies are still striving to beat all kinds of cancers. Next up, we go to OneCell Diagnostics, a company that gave presentations and had a poster at AACR. Before the event, the company announced the launch of OncoInsights, a multimodal cancer diagnostic panel, and it also announced a partnership with BioScribe Genomics. To tell us about all of those things is Mohan Ottawa, CEO and co-founder of OneCell Diagnostics. So I wonder if first you could maybe go through some of the history of OneCell Diagnostics.
Sure, Jim. OneCell Diagnostic got started about three and a half years ago. It has a little bit of history because I was an investor in another company, angel investor, that was doing a single cell CTC, first generation. And I and Dr. Gover Shafi, who is our Chief Medical Informatics Officer, were working on a company called Index AI, which was focusing on multi-omics data analytics. And the history of that started with our collaboration with Parker Institute and 6 of their affiliates, the Stanford, UCLA, UCSF, Sloan Kettering, MD Anderson, and UPenn, in order to facilitate immunocancer therapy translation research, accelerating it. And the hypothesis there was that the field is moving from single omics to multiple omics. And, in oncology, one shoe doesn't fit all. So you have to personalize the treatment. Finding the right treatment for the right patient at the right time with the right dose is a challenging task. But the impact of that is significant. This is where I think integrated software approach and AI comes in very handy. So we were pursuing that path. And we were working with some of the pharma and biotech companies in addition to the academic centers. And then it was pretty clear that the field is also moving from bulk sequencing, so next generation, and I might be making it a little more complicated, but this field of personalized medicine is moving from bulk sequencing, next generation sequencing, to single cell. And one thing led to the other, and we decided to sort of integrate all our efforts to create an integrated platform for accelerating precision oncology. There is a deep science part and there is a deep technology part. And the action is at the intersection of deep science and deep technology to deliver personalized care. That's how OneCell got started about 3.5 years ago. Index AI has been there for past 6-7 years. Okay, and then I was an investor in another company, and so we have 4 co-founders. Dr. Jayant is an avid scientist with a very deep knowledge of chemistry and biology. Dr. Gohar specializes in the deep AI-enabled multi-omics technology, and the combination of that is delivering groundbreaking results.
Could you perhaps explain what OncoInsights are?
So OncoInsight is the name of our product. It is a test we are offering. It's called LDT, Lab Developed Test. So our lab has a CLIA CAP certification. We are based here in Foster City, California. The CLIA CAP certification means the lab has certain stature, certain standards. It follows certain protocols. And the test that comes out of it has certain minimum requirements. So early stage technologies, which hasn't gone through an FDA approval yet, the next best thing is a CLIA CAP certified lab offering a test. And this test, they go through rigorous testing, rigorous validations. It is governed by board certified pathologists, oncologists. So we have and it's also done by CLS, the CLIA certified lab people. And so we are offering this test. What OncoInsight does, it has simplified the complexity of all these multidimensional data analytics. We provide the kits to collect the sample. It has 2 tubes, 10 ml each, simple blood draw. Patient will go through the blood draw. Some phlebotomist will visit their homes or whatever. They can go to the draw station, and those are shipped to our lab. We process those blood samples. We take, the DNA, the RNA out of it, and we create libraries, and then we apply our testing or picking up the circulating tumor cells. What happens in the cancer is that a gram of tumor may shed about 1 million cancer cells a day in the bloodstream. It is these circulating tumor cells, those are the culprits. OncoInsight has developed, it's a test, and we have developed the technology to capture this 1 in a billion circulating tumor cells from those blood tubes, the test tubes that we get the blood from. That's the groundbreaking science that we have. OncoInsight test conducted in our lab, the first thing we do is capture that 1 in a billion circulating tumor cell. Then we isolate those cells. We do it with a high degree of precision. The groundbreaking science that has been developed by us, which are proprietary IP, is order of a magnitude high degree of sensitivity in capturing these circulating tumor cells. Then the purity of tumor cells, order of a degree higher. And the simplicity of the capture is order of a degree higher. So if I take these 3 axes, OncoInsight is 1,000 times better price performance than anything that is out there. These are the cells that they go from one place to another place. So you may have a breast cancer patient. The tumor in the breast will shed these cells. They go and settle somewhere else. And then they start growing there, building that, the secondary tumors. So we have to capture them at an early stage, and then we have to characterize them. Okay, so after the isolation, we amplify, we take their DNA, RNA, and then we do the next generation sequencing. And we do DNA sequencing, RNA-seq, we do proteomics. These are all different scientific fields. And then what we have in OncoInsight test is a software that applies AI and ML to analyze all this data that gets generated from those two test tubes of blood and from those circulating tumor cells. And then we have models to predict that tells the oncologist what medicine will work for that patient, what medicine may not work, or what medicine may work for a period of time, but after 6 to 9 months, it will develop some resistance. So this is where we are working on a predictive model and helping oncologists make the right decisions for that given patient. It's a complex back end, lot of science, lot of technology, but the bottom line is that we produce very nice reports that will help oncologists make critical clinical decisions for a given patient at that given time point. And that will change the course for that patient.
You already mentioned some of the advantages that this technology has. What are some of the other advantages over existing methods?
So, Jim, if you look at historically precision oncology, that is the future of oncology management. The first generation of precision oncology came from tissue and genomics. Good companies like Foundation, they made their mark in the first generation. But tissue is often an issue. The biopsy is a surgical procedure, that patient has to undergo. So second generation came liquid biopsy. That means from blood, you can do this testing. And that became very popular, and very good companies, like Guardant and Natera made their mark. But the current liquid biopsy companies, Jim, they are all using cell-free DNA or a ctDNA. Those are fragments of DNA that get spun out, from the dead cells. Cells die. That's a normal course. The tumor cells, shed the ctDNAs, fragments of DNA. And so that's a good biomarker. It tells you a few good things, but it doesn't tell you the whole picture. So first generation was tissue biopsy. Good, but it's difficult to get more and more tissues. The second generation is liquid biopsy. That's only telling you information that is in the DNA. OncoInsights, our test, we are developing the third generation. We call it cell biopsy, which is capturing the circulating tumor cells. And live tumor cell has at least 2 orders of magnitude more information than the DNA, ctDNA. So your question, what is different? We are now have a lot of information from that blood. We call it the cell biopsy, the live tumor cells, and we can do DNA, RNA, proteomics, transcriptomics, metallomics. That is not possible to do on DNA. Proteins on the surface of the circulating tumor cells give you a lot of information. That is not possible in the DNA. And our hypothesis is that what we call the cell biopsy, which is the third generation of testing, is all encompassing. Our hypothesis is that ctDNA information, when you integrate with CTC DNA, RNA, proteomics, the CTC multiomics, when you integrate that, you have a much deeper insights into what's going on in the body of that cancer patient. And we present that information in a very structured form to the oncologist, who then can prescribe the right medicine.
What does this mean for biotech and biopharma companies? And I guess at the end of that line, what does it mean for patients?
Good question. Well, first of all, in United States, we are primarily targeting the early adopters, and early adopters are biotech and biopharma companies that are trying to bring out new drugs, and they do clinical trials. The early stage clinical trials, there is a phase of translation research where they're trying to figure out what are the right patients to be selected. So selecting the right patients for a clinical trial, deselecting the wrong patients, therapy response monitoring. Once you give the early doses to the patient, is it working? Is it not working? If it is working, why it's working? If it's not working, why it's not working? So therapy response monitor, minimum residual disease, right? These are all informations very critical for accelerating the drug discovery, drug development, and the testing through the clinical trials. Before, this information wasn't available. So what you had to do in a traditional route was that you give the medicine, you wait for 3 to 4 months. Let's say you're doing immunocancer therapy drug. The only way to know whether it's working or not working is through what is known as RECIST criteria. Now, RECIST criteria work on the in vivo imaging, the CT scan or MRI or PET. That takes 3 months. Then only know, if the tumor is shrinking, that means my drug was working. Now, 3 months are gone, and time is money for these pharma companies. So instead, what we do, within a matter of 10 to 15 days from the dosing, through Onco Insights, we can tell the pharma researchers, the chief medical officer, the chief scientific officer, the biomarker discovery guys, the people working on that cancer research clinical trials, that, it's not working. Because let's say if you're doing immunocancer therapy, and let's say it's an CTLA-4 test, which is showing the PD-L1 protein on the surface of the CTCs. So we can tell them that information instead of waiting for 3-4 months within a matter of a couple of weeks. That saves time and money. Not only that, the deeper insights we provide to them, the different genomic information, proteomic information, that can also give them the indication of why it is not working and what could you do. So if a clinical trial, they call it a basket trials, where multi-arms, they can choose and pick and choose and, switch the arms and really see what is the optimal stuff. So throughout that journey in the clinical trial space, we leverage a lot of AI, and the tests like Onco Insights will provide them much, much better information to accelerate the trials, reduce the time, optimize the chances of success, give them the better understanding of what can and cannot be done. So that's for the biopharma companies. This will perhaps go into what we know is known as companion diagnostics. So that means when the drug is available in the market, to know for sure that this drug will work, our test done on the patient will give them a higher degree of confidence whether that drug will work for the patient or not work for the patient. So that ability to do that prediction is very helpful to the patients.
And could you tell me a bit about the partnership, though, the recent partnership with BioScribe Genomics and what the implications of that are?
Good question again. Cancer is like an elephant and 6 blind men. Depending on where you touch, you describe that elephant. New science is evolved very rapidly. The field of genomics, the field of transcriptomics, the field of proteomics, pathology, radiology, this imaging. But each one of them are in siloed. Radiology and pathology are two sides of the same coin, but they don't talk to each other. That talking happens in the head of an oncologist when they look at two reports. And no one modality gives you the complete picture. Time has come to integrate the information from all these different modalities: genomics, proteomics, transcriptomics, metabolomics, and integrating that, abstracting it, analyzing it, and then coming up with this thing. So that's where the field is moving. Today, we capture from a blood sample maybe 5 to 15 cells. And today we use some of them for genomics analysis. Some of them are used for proteomics. Some of them are used for transcriptomics. What BioScribe has done is build some kits so that on the same cell, you could get information which is genomics, transcriptomics, and proteomics. So it really helps us, this partnership, first in its class, first time in the world, a true single cell captured from patient's blood, captured with a live single cell, and being able to do multi-omics on it, the single cell genomics, the single cell transcriptomics, the single cell proteomics. That's what this partnership enables us to do on the same cell, as opposed to two different cells doing two different things. And once that data comes back, OneCell has developed AI-enabled deeper data analytics to present a report that will help oncologists make those critical decisions. We don't make the decisions. We do data analysis, we do data modeling, and we do recommendations. But the final decision is always made by oncologists because they have deeper insights.
And I guess that with the way that technology is evolving and advancing so quickly, it must be a very fast changing field.
Absolutely. Jim, the field is changing very rapidly. Like I mentioned to you, from bulk sequencing, it has gone down to single cell. From single modality, it has gone to multi-omics. AI is playing a very important role because the amount of data that gets generated from a single cell level is humongous, and no human being can make sense out of it. So that's where, again, AI comes in handy to aggregate that data, abstract it, and be able to apply the predictive models to make the recommendation. At AACR, we had our poster, and there the field is moving so rapidly, a lot of new research coming up, so I think it's difficult for practitioners to keep track of it. And Jim, that's again where, we have developed some interesting software applications to assist and guide them.
You just mentioned AACR. How was that meeting? Were you presenting, or was it just the poster?
Yeah, we were presenting. We also had a poster. We did a joint, event together with BioScribe, and it was very well received. Before, people were not very sure what would you do with single cell. It was only in research. Between last AACR and this AACR, we saw a significant increase in the interest level in the single cell. So clearly the field is moving from bulk sequencing into the single cell arena. So it was heartening to hear, and see the level of interest quadruple from previous event to this event in the single cell arena.
Was there anything else that caught your eye at AACR?
Yeah, I think the antibody drug conjugate is another field of interest. For the past 10 years or so, immuno-oncology was always the darling of the crowd, and it still is. But, we certainly saw an interest picking up with ADC, antibody drug conjugates. It's a different way of delivering medicine to the cancer patient. I think that field is now looking very, very interesting.
So what are the next steps for your company now?
We have now a lab set up. We have the CLIA/CAP certification. We have the assay. We started generating customers now. We have signed up a few new customers, some biotech, biopharma, and some pharma customers. ADC is our focus. We believe very strongly that number of companies entering into ADC space and number of drugs that are in the clinical trials. And one unique thing about it, Jim, that OncoInsights, our assay, is unique. No one else is able to provide proteins. When I say no one else, I mean the traditional liquid biopsy companies. They are dealing with ctDNA. They don't have the ability to do the RNA or the protein. For ADC segment, you need proteins. So we have a unique position able to help those pharma biotech companies. And that's our focus area. And BioScribe partnership was very helpful in getting multiomics on a single cell, particularly the proteomics side, which will help the pharma biotech companies. So jointly, we are targeting, those companies that are doing the early stage translation research in ADC space. One thing that is very clear is the overwhelming amount of data that gets generated in genomics, transcriptomics, proteomics. Without the use of AI, and ML, it's not very easy to make sense out of it. So we're taking a multi-pronged approach. We have developed a lot of convolution neural network models to analyze that data and to do the prediction, of the outcomes. That's number one. The second thing we have done, Jim, is how do that information gets presented to both the oncologist who are making those critical clinical decisions, but also how does it help patients to understand what's going on. So we have developed an application, we call it iCare. It's a precision oncology console. Think of it, as a co-pilot. See, the medical fraternity, oncologist, this field is never taught in the medical school when they went to medical school. So it's very new. And they are very busy people. They don't have time to learn about all the presentations that happen at AACR, at ASCO, at ESMO. What does that mean? A certain new gene identified, certain protein expressed is going to make a difference between whether the medicine will work for a given patient or not. So all this stuff we are using AI and particularly Gen AI to give that information at their fingertips. If they find certain things that they don't know, with a click of a button, it tells them, and with the right context of that patient. So we have created this beautiful precision oncology console and a genomic co-pilot that aids them every step of the way in ordering the right test, getting the right report, interpreting the right report, and prescribing the right medicine. That's on the doctor's side. The same thing we have done on the patient side, Jim. We created an application called OSAKI. Saki is a Sanskrit name for a dear friend, where you can ask any question, no bars, no barriers, right? And it will give you the answer to the right context. So a lot of cancer patients, they don't understand this genomics, proteomics, this, that, other. They go through the treatment without knowing and without being able to understand the report, without being able to understand what's going on. So we've created this avatar to help those cancer patients answer their questions and be able to monitor, their health. So we've taken a very holistic, integrated approach, and we're going after in a very subtle way to really, make our dreams come true, which is to democratize precision oncology, making it accessible, making it actionable, making it affordable, so that every oncologist in every hospital, in every corner of the world, should follow the precision oncology practice. And the innovation for affordability has helped us bring out affordable tests so that no patient is left behind.
It's clear just how much the evolution of technology has made to companies looking to treat all kinds of conditions and diseases, including cancer. And certainly a whole new vocabulary has developed over the last 10 to 15 years. Another firm that attended AACR and presented data there is Microbiotica, a company dedicated to tackling diseases, including cancer, through making changes to the microbiome. To tell us about the new data that was presented and how the microbiome is important in treating diseases is Microbiotica's Senior Vice President of Research, Matt Robinson. So I wonder if first you could tell us about the journey so far from the early days of the company's founding and the reasoning behind that to where we are today?
Yeah, certainly. So Microbiotica span out of the Wellcome Sanger Centre back in 2016. Trevor is principal investigator at the Sanger Centre. He is still our CSO and still a principal investigator at the Sanger Centre. Trevor was one of the sort of founding key scientists in the microbiome space. One of the early things he did was he cultured out a number of the bacteria in the human microbiome to help map it. And at the time, the human microbiome, most of it was thought to be unculturable. And scientists like Trevor and others disproved that. So Trevor span out the company with the idea of industrializing those processes to create a bank of bacteria cultured from the human microbiome for two things. One is that will give you a bank of potential therapeutic bacteria, but also by using the genomes of the bacteria, when we profile the microbiome in patients, we do it by genetics. By having all those genomes, that gives you the blueprint for the microbiome. So the more genomes, the better the coverage of those genomes, the more precise and accurate the analysis of a patient's microbiome is. So it's really with the aim to get to a clinical level resolution when we analyze a patient's microbiome to be able to create therapeutics and diagnostics.
What are oral precision microbiome medicines?
We use our platform to profile the microbiomes of patients from clinical studies. And with our platform, we have the resolution to really identify the key bacteria that are driving or involved in that disease process. And so we think that key resolution, that precision enables us to identify those bacteria, which that we can make into products. And that's what we often do, or we can make it into a diagnostic to understand, patients which have a bad microbiome, which ones have a good one, etc.
Following on from that, then what are live biotherapeutic products?
I mean, that's really a regulatory term. It's a class of, therapeutics that are live products, so they're bacteria or viruses or something similar to that. So in our case, when we talk about live biotherapeutic products, what we're talking about is small consortia, 8, 9 or so bacteria, so normal gut commensal bacteria mixed together. So they're freeze-dried, mixed together and put into a capsule. That's how we deliver them.
And what kind of conditions and diseases can you tackle through this kind of process?
The list of diseases now that have been associated with the microbiome is huge, and it's ever growing. So the microbiome is a component in just a huge list of diseases, from autoimmunity, Parkinson's and other neurodegenerative diseases, a lot of the metabolic diseases, type 2 diabetes, some of the liver diseases, all strongly associated with the microbiome. So understanding how the microbiome contributes to these diseases and then how we can therapeutically treat it is a key thing. So, we think this has a huge potential across a wide number of diseases.
So could you maybe go through some of the links between the microbiome and disease and how treatments affect the microbiome and alter it?
Yeah, so I guess our best way to treat at the moment is through FMT. And there's some really key wins there. So FMT is when you take the microbiome from a normal, healthy individual and give it to somebody with a disease driven by the microbiome and try and normalize their disease. That's a nice way of saying a poo transplant. That's been used in a number of cases. It's widely used now in C. difficile infection. So that's an enteric gut bacterial infection that is primarily hospital-acquired infection of immunocompromised patients with a very odd, a non-diverse, a very simple microbiome that would look very unusual compared to ours. So C. difficile really thrives in those sort of conditions. And it can be treated really well by antibiotics. But the problem with the antibiotics is they make that odd microbiome even worse. And the C. difficile can form spores. And so it comes back. And the more you treat it, the worse the microbiome gets. So actually, if you treat it with antibiotics and then give them FMT, they do so much better than if you just treat them with antibiotics, especially those recurrent patients where it's recurring and recurring and recurring. So that's an example where something like FMT works really well, because what you're doing is you're taking a really altered microbiome and you're just normalizing it. Whereas many of the conditions that we look into, like inflammatory bowel disease, particularly where an area we're interested in is immunotherapies in cancer, seem to be linked with the microbiome. And that's been shown because we've done it and others have shown this, that the microbiome of the patients before they receive therapy in some way determines whether or not they respond to that therapy. The microbiome is known to be really intimately linked with your immune response. That's why it's linked with so many of these inflammatory diseases like autoimmunity, etc. And so what we see there is the microbiome is slightly altered from a sort of healthy individual, but what we see is there's sort of signatures of bacteria, key species of bacteria that are differentially expressed in the patients. So in the case of those that respond to immunotherapy, there's different bacteria in the patients that respond and don't respond. But you can see that across a number of different diseases, and it sees impacts that even many people that have done studies didn't understand. A good example is there was an FMT study in autism spectrum disorder, and it was done in autistic patients with constipation to help with their constipation. But actually, a lot of their cognitive functions improved as well. And we know that there's a difference in the microbiome between autistic people and normal, healthy individuals. So you see here that the microbiome can impact many different features of many different diseases with inflammation.
How does the fact that everybody's microbiome differs and even fluctuates on a fairly constant basis, how does that affect both the disease and treatment response? And I would assume that that would maybe even affect things like clinical trials.
Yes, exactly. And it does make it really challenging. And that's why a high resolution and being able to analyze patients' microbiomes is really important, so that you can see across geographies the same sort of effects. You see in the same bacteria coming up. So that's really important. And it speaks to our strengths in that level of analysis. Yeah, as I said, sometimes it's big changes in the microbiome, sometimes it's more subtle changes in the microbiome. So it's a challenge to the field, but what you need is really good quality clinical studies to understand what the key important bacteria are.
Can it also be used for diagnosis?
Not at the moment. I don't think I know of a test that's out there. There are a number of companies that will analyze your microbiome for you and provide you with some guidance, but it's not used as a diagnostic in the healthcare setting as such yet. We see differences in patients and it's consistent. We're a little way off, I think, of getting that into a clinic-ready diagnostic physicians can make decisions on, but it's coming. We're getting closer and closer to having that sort of resolution here.
I think a lot of people, especially recently, have associated the microbiome with products that can improve your health and almost a miracle cure. Has that been a negative on the industry?
Yes and no. We see a lot of enthusiasm from patients around what we're trying to do. So that's a real positive. It's undoubtedly helped where we've gone into the clinic with our 2 lead projects. We're seeing enthusiasm from patients to come on and try these sort of things. But I think anywhere where you get a lot of hype and then it doesn't follow through always creates some headwinds. And some investors we know are reserved about going into this area. Some pharma are quite reserved because, again, they went in, but maybe too early. And some companies that were rushing to try and make products rather than taking their time to do it the right way.
Regarding your own work, what's MB097 and how does that work?
MB097 isn't an obvious one on the face. So it's using healthy gut commensal bacteria to help treat melanoma, skin cancer, and advanced melanoma at that. So it's not an obvious one. Some seminal work came out in 2017, 2018 by a number of different groups almost simultaneously that said the microbiome of patients before they take these immunotherapies that use your immune system to attack the cancer and remove the cancer, that the microbiome of the patients was key to whether or not they responded. And so what we did was we took 4 independent melanoma studies, one of which was a collaboration of ours with Adam Brooks Hospital here in Cambridge, UK. And what we did is we analysed the microbiomes of those patients and looked across the 4 studies and found bacteria that were in common across the 4 studies that are associated with a positive response to immune checkpoint inhibitors. That's the immunotherapy that works particularly well in melanoma. I mean, in terms of metastatic melanoma, these checkpoint inhibitors, these immunotherapies have been transformative. The 2-year survival of somebody with metastatic melanoma going back 10 years was very low. It is, down to about 5%. Now, with this class of drug, about 50% of patients are in what we call long-term remission. That's up to 10 years now. They've been measuring that. So a truly transformative new modality came into this metastatic melanoma and just changed the landscape for patients unbelievably. But what that means is half of those patients are left with the old traditional therapies that haven't been working. When we looked at the microbiome patients, when we looked at those patients that didn't respond, about 80% of them had a microbiome that we thought was non-permissive to the response, so it didn't help them respond. So the aim of MB097, and what's important is what we saw primarily is it was a lack of bacteria associated with response that was driving this rather than bacteria that were associated with non-response. So it wasn't bacteria in non-responders that were stopping them responding. What it was good bacteria in people that were responding primarily that was enabling to respond. We looked at this in many different ways and we honed down onto these, what we think are 9 key species, and that is essentially what is in MB097. They're lyophilized, they're grown up in fermenters and lyophilized and put into a capsule for patients to take. And all we're trying to do is increase the abundance of these bacteria in the microbiome of patients so that they can go from being a non-responder into being a responder. And what's really, I think, key and exciting for here is a couple of years after we started this project back in about 2021, there was a couple of papers that said they took advanced melanoma patients that had failed to respond to checkpoint inhibitors, so these non-responding patients, they gave them FMT from patients that had responded to checkpoint inhibitors, and a number of those patients were then able to go on to respond to checkpoint inhibitors that they hadn't responded to before. So this was really key, really demonstrated to us the potential here. The problem with FMT, it's hard to scale up. It's not very palatable for patients. It's difficult to source from patients. And when you have to have patients that have already responded to therapy to be able to do that, that's a really challenging thing. And there's a bit of hit and miss. There's the FMT world is full of anecdotal data of super responders, super donors. So certain donors are better than other donors. And it's really hard to get a grip on that. What we're trying to do is essentially just take those key components that we think are driving that beneficial response in the FMT and giving it to patients. And the other advantage we have is we're dosing this daily, so we can keep that. Whereas FMT, it's very difficult to repeat dose.
You recently presented some results based on this. Could you tell me about those?
We identify our bacteria in a clinic first approach. This whole interaction of the microbiome with its host, or in this case with patients, we understand some of those interactions. So we know some metabolites produced from bacteria have this direct effect on the host. We know that other metabolites have this effect on the host, etc. But actually, it's such a complicated interaction. We're only just beginning to understand that. So we're in this situation where we've got these bacteria that we think are beneficial for patients and we want to give to patients and drive a good response because there's clinical association, but we don't know how they do it. So we spend a lot of time once we've identified the bacteria we want to do to prove that these bacteria can help a patient. And so we've used mouse models in this case, but we've also used a lot of human in vitro assays using human cells and our bacteria to understand how our bacteria can interact with that human immune system and drive that immune system to kill cancer cells. And we know the mechanisms by how these checkpoint inhibitors work. So they work through specific immune cells that kill the cancer cells. And what we can see is our bacteria in MB097 really enhance that tumor killing potential of these cells. So we see how it can synergize with immunotherapies to help kill the cancer of patients. We've understood this for a while, but now we're really starting to dig down into the details of this is happening. And in terms of the immune response, what we showed recently was some of our bacteria are able to activate a particular cell called the dendritic cell of the immune system. It's one of the sentinel cells that sits in the gut, and its job is to direct the adaptive immune response. And that's the immune response that kills cancer. So we've been looking at how these cells drive that adaptive immune response, and they release a really key cytokine, a protein called interleukin 12, that is a key potent activator of these cells. So we think we're starting to understand how our bacteria really work at the molecular level in terms of driving these responses in patients. What we've also shown recently is not only do they have this direct effect on dendritic cells driving this response, but they release metabolites that work directly on these cells that kill tumor cells as well. And you can imagine that works through the bloodstream and so can work distantly from the gut. So maybe activating these cells within the tumor as well to help drive. So we have a consortium of multiple bacteria that we think all interact with the immune system in slightly different ways or overlapping ways to drive this immune response that then goes on, has a beneficial effect, hopefully in patients.
Often these treatments are given when somebody isn't responding. Once somebody's been diagnosed with melanoma, would it be useful to just give this to everybody that's diagnosed with it as soon as possible so that it improves their response?
Yes, we believe so. It's the easy answer to that. The key, obviously, with a cancer patient is do no harm. And these checkpoint inhibitors are absolutely, the best hope the patients have for a durable response. So we and others in the field have been nervous about going first line on top of these checkpoint inhibitors until we've shown that they have some clinical efficacy and clinical benefit. And so that's why the FMT studies were initially done in patients that had failed to respond. People have now gone on to do FMT studies in that first line setting with checkpoint inhibitors and shown that you can increase those response rates in patients in that first line setting. So we're really encouraged by that, and we're really excited by that. And that's where we would like to take our therapy, because that's where we can, obviously, the earlier and more strongly you can treat a cancer, the better the outcomes. So yes, that would be our vision, is that's where we would like to go to. But at the moment, our initial trial is in that non-responder patient as well.
What's the reaction been like to your results?
So we haven't got the results of the clinical trials yet. Our first trial is ongoing.
Okay.
So the posters and what we've been telling people about is really understanding the mechanisms of how our bacteria, we think, interact with the human immune system and drive that response. We're doing it from an understanding our drug point of view, but that mechanism's not been known and recognized in literature. So it's a new scientific finding for the field as well.
You've just had a new press release on MB310. I wonder if you could tell me a bit about that.
Yeah, so inflammatory bowel disease is a chronic disease that is a real struggle. My father had it all his life. It is intimately linked with the microbiome of patients. And interestingly, it's quite a flaring disease. So you can have periods of where you're in remission and then you flare. We think that might be to do with patients' microbiome changing over time, leading to triggers and to enhance the disease. And there's basically two forms of inflammatory bowel disease. We've been particularly interested in ulcerative colitis because, again, FMT studies there have shown that it can take patients with mild to moderate disease and put them in remission. So it can have great therapeutic benefit. MB310 is designed slightly different in the sense that we actually looked at an FMT study and we looked at what bacteria are engrafting from the donor into the recipients and were really linked with clinical benefit in that scenario. And so, again, we identified a consortium of bacteria that we think has real potential in ulcerative colitis to be able to drive these patients into remission. But because we can dose this regularly, unlike FMT, we think we can keep them in remission as well. So we're super excited about 310. We think it's going to have a lot of benefits for patients. Like MB097 in melanoma, we've been working on these bacteria that are linked with benefit in ulcerative colitis and understanding how they work. And it's a similar story. So there we have 8 healthy gut commensal bacteria. And what we see is they interact with the host at multiple different levels. So you see quite often the epithelium is degraded. Our bacteria help repair that. They also interact with the immune system in multiple different ways that we think will be beneficial to the inflammation that we see in inflammatory bowel disease. And so, we're releasing data on MB310 to show the mechanism there. And it's a similar concept, different bacteria having different effects on the host. So this is quite different to your traditional therapy where you block a pathway either with a small molecule or an antibody, or we're having multiple different effects on the host. So we think where you get resistance to traditional therapies, that's much less likely with a microbiome therapeutic.
And in both of these cases, where are you at with clinical trials? Where are you on the pathway, I guess?
So they're both in their first in human clinical trials at the moment. So these are phase 1b studies. Because the microbiome therapeutics has a really good track record of safety, etc., we're doing our first time in human studies in patients to check it's safe in patients. They have a slightly different microbiome to healthy, so it's the most relevant patient population. But we also hope to have some good clinical benefits in these studies as well.
And in terms of other things in the pipeline, you mentioned the connection between the microbiome and lots of different conditions. Are you looking at other things?
We're a small biotech, so we have to be focused and direct. We believe in, really doing the science properly on everything. And that's where the microbiome's failed in the past. So you got to do deep science. So we are quite focused as a company at the moment. So we have a study ongoing. The sample collection and the study itself is funded by Cancer Research UK. It's with Pippa Corey, Adam Brooks Hospital, that we did the previous melanoma study with, but it goes across multiple indications and multiple treatments to understand how the microbiome affects these cancer patients on immunotherapy. So we expect to use that sort of data and other data to be able to expand out of melanoma into other cancers and to treat other cancers. And we're also very interested in following up our UC product by looking at other inflammatory bowel diseases such as Crohn's disease, but also autoimmunity and things like that.
I did quite a lot of articles on the microbiome while covering the food B2B industry, so it's clear that the volume of knowledge on the microbiome and its role in so many things is still growing. 3 very interesting guests this week. Actually, we had a 4th conversation with a company attending the AACR, but just not quite in time to edit it for the podcast this week. So we'll start next week with an AACR part 1.5 maybe. If you have questions or comments or suggestions for guests, please do get in touch, and you can do so at jim@deeptechdigest.com. So until next week, wherever in the world you are, I hope you have a great week. Thanks for listening, and I hope you'll join us again next time for another Base to Base Biotech podcast. The Base to Base Biotech podcast is an Air Coastal Media Limited production.